Hi, this video is about the group of biomolecules or macromolecules known as proteins. There we can see hemoglobin, a protein we'll be talking about in this lecture. Now, proteins are the working group of the biomolecules. They're involved in the trades, security, energy, construction, deliveries, maintenance, upkeep. Other core functions of living things are carried out by proteins. You could almost call them a blue-collar group of molecules. Now, to get technical, proteins are a polymer of amino acid monomers. So amino acids are the individual small molecules that make up larger proteins. An amino acid can be joined together one next to another into a long chain, and that chain will eventually fold into the shape of a protein. There are many separate amino acids occurring naturally. About 20, give or take, are important to an introduction to biology class. Here we can see the structure of an amino acid. There is an amino group on one side, and on the other side is a carboxyl group. In between is a carbon. We call it the central carbon, and sticking off is a hydrogen, and on the other side is what's called an R group, or a side chain. The central carbon, the hydrogen, the amino group, and carboxyl group are consistent and found in all amino acids. The R group varies. There are many different R groups. This is what makes an amino acid unique and separate from the other amino acids. So here we see all the different amino acids that we might run across in a more advanced biology class. You don't have to know these now in an introduction to biology, but you can see that the R group gives each amino acid its own characteristics: pH, polarity, attraction or repulsion away from water, and or electrical charges can be found. So the shaded parts of all these different amino acids are unique to the different kinds, and some are polar and they're attracted to water. Some are non-polar and they would be repulsed by water and move away from it. Each amino acid interacts with the other amino acids in its own way because of this. The non-polar R groups would make things hydrophobic, and so they would cluster together avoiding water. And the polar groups are hydrophilic, meaning they're attracted to water. So each protein has many, many different amino acids, and it could have some parts that are attracted to water, being polar, and some parts that are not attracted to water, being non-polar. That combination of hydrophobic and hydrophilic makes them amphiphilic, a little bit of both. The amino acids in a protein determine its structure and its function. There we can see a chain of amino acids, and as a protein is built up and processed, it leads to a more complicated three-dimensional shape. Now, we can talk about peptides and peptide bonds. This comes from the Greek word peptos, which was ancient Greek meaning to digest. Dehydration synthesis, also called condensation reactions, creates a peptide bond that joins together amino acids. So a peptide bond is a bond between two amino acids. We can see it's called dehydration because water is coming out of it. Here's one amino acid and another. From the carboxyl group of one amino acid to the amino group of the adjoining amino acid, a hydrogen, another hydrogen, and an oxygen come away, leaving water, a byproduct of this reaction. But now we've bonded two amino acids together. A peptide can be called a growing polymer of two or more amino acids. This is a covalent bond. They're sharing valence electrons. So peptide is a special name for bonds in proteins. And then, remember the R groups are there too, because they'll impact what happens after the proteins are linked together in a peptide bond. We can also apply prefixes to the number of amino acids involved in the bond. So dipeptides have 2. Oligopeptides have fewer than 10 to 15. more than 2 or 3, but not a huge amount. Then finally, polypeptides have more than 10, and proteins have many, many more. Here we see oxytocin. It's a protein made of about 9 amino acids. We can see them color-coded here. 1, 2, 3, 4, 5, 6, 7, 8, 9. And you'll notice there's another feature of proteins here in oxytocin. In this amino acid-based protein, there is a bridge from the sulfur molecules on one R group to the sulfur molecules on another R group. This starts to show us how the chain of amino acids can begin to bend and fold because of the components of the R groups. Oxytocin also plays a role in social bonding, reproduction, and childbirth. And so it's a protein made of amino acids. It's an awfully small protein with only 9 amino acids, but it's very interesting that this is a chemical that affects subjective or abstract qualities of life, such as emotional trust and suppressing fear and deciding who you're friends with and who is family. It's a chemical in our bodies that is released during childbirth and shortly after childbirth. It's been called the love hormone or the trust chemical. So from these basic chemicals, we get big ideas about what it means to have community, and those are harder things to measure, but they're influenced by chemicals in our body, like this hormone. A little bit more about oxytocin, a basic protein. Part of all life is homeostasis, and the oxytocin that is released during childbirth is an example of the process of homeostasis because it is released in what's called a positive feedback loop. Some oxytocin goes to changing the body, and some of it works as a chemical that goes back to the origin of the oxytocin, and the positive feedback says some of it leads to signals to make more of it, and that increases, and your body is flooded with oxytocin until it reaches a point where a negative feedback loop stops or slows that process down. So we can connect our learning about proteins to bigger processes that affect all life, like homeostasis. Now, proteins are organized in their structure. There are 4 levels to the structure of a protein: primary, secondary, tertiary, and quaternary. Here we see the primary level. We can just talk about the sequence of amino acids, which one comes after the other. Just like letters can form words and words can form sentences, the sequence of words can give a different sentence a different shades of meaning. So the primary level of a protein is what are the order of the amino acids one after another in a chain or polypeptide. Beyond that, at the secondary level, the bonds between the amino acids push and pull the structure into 3-dimensional shapes. Some common shapes we've seen are alpha helices or beta sheets. And so you can see these more advanced shapes start to show up as the individual R groups in proteins push and pull against each other. It gives common shapes. Some of the molecules are working to avoid water, or some of the molecules are working to be attracted to water, and that takes the normal sequence, the chain, and folds it and pulls it. Then the tertiary, or third level of structure, leads to further folding and coiling. The R groups do more interactions. The larger structure, the alpha helix or the beta sheet, attract or repel each other. Hydrogen bonds start to show up, and S or sulfur bridges, as we saw in oxytocin, show up. And then remember, this whole thing is sitting in an environment, and so a more acidic environment could cause the protein to have one shape. A more alkaline environment could cause a protein to have a different shape. Now, the tertiary level gives us a single protein molecule. We pretty much have a protein there. Sometimes for some proteins, it goes further into the quaternary level. There, associations of several different peptides build a protein up into a protein complex. Here we can see hemoglobin, a common protein, and it has 4 subunits. 4 separate polypeptide chains have come together. So, let's talk about hemoglobin a little more. There's 4 separate polypeptides at the quaternary level. In this computer-generated image, we can see some of those parts of the secondary level, the helices that form. Of course, it has the job of carrying oxygen in most vertebrate animals. There are some other varieties of chemicals that carry around gases, but we're not going to talk about those. Red blood cells are so full of hemoglobin protein that they actually break down or dismantle their nucleus to make room for even more. The main job of our red blood cells is to be packed full of hemoglobin proteins to carry oxygen from the lungs through the heart out into the different cells of the body. We can also talk about why the primary structure of a protein matters. There is a disease called sickle cell. It leads to red blood cells that have a sickle shape. That's a cutting implement that farmers and gardeners use sometimes. So, sickle cell is caused from a change of just 1 amino acid in hemoglobin, but that affects all the hemoglobin in the red blood cell. And then beyond that, many red blood cells affected by sickle cell affect the entire individual. A whole organism feels the effects of these altered red blood cells. There's a genetic component to sickle cell, and there's many symptoms from it, notably pain in the joints. And strangely, sickle cell gives partial resistance to malaria. So, the primary structure of a protein matters. Changing just 1 amino acid is like changing 1 word in a sentence that changes the whole meaning of the sentence. In sickle cell, it changes the shape of hemoglobin, and then the red blood cells that hold hemoglobin. And beyond that, there's effects on the entire individual. Now, this ties to another common characteristic of all living things, namely evolution. This map shows us where malaria is commonly found in the world. Many parts of Africa and also parts of Western Asia. The places where the genetic component of sickle cell is found also overlaps with those same areas. So, because sickle cell gives some resistance to malaria, in some ways it is an adaptation to resist the survival pressure of dying from malaria. Sickle cell is not a great thing to have, but malaria is a terrible disease to have that kills millions of people every year. So, it's the idea that the enemy of my enemy is my friend. If sickle cell is bad for you, that's a problem. But if it helps you survive malaria, well then it's not as bad as it could be. So that ties sickle cell and hemoglobin back to evolution. Here's a different protein. This shows the primary structure of insulin, the protein involved with regulating blood sugar levels. There are 2 polypeptide chains, and they are connected together by these sulfur bridges. There's 51 amino acids involved in a single insulin protein. Now, at this level, you wouldn't have to know all these amino acids, but you should realize if I say this is a protein, then you should say the small monomers that make it up are amino acids. And so these 3-letter abbreviations, even if I don't know what it is at this point, it must be some kind of amino acid. Another thing about protein structure is that proteins are not necessarily rigid. Many can change their shape or their conformation. For example, proteins that serve as opening and closing gates on the membranes of cells need to change their shape. A theme to all life is that form fits function. In this case, the 3-dimensional structure of a protein, which is based in that order of amino acids, determines its job that it can do, or its function. Now, the opposite of the conformation or the putting together of the protein is denaturation, a drastic change that destroys the protein shape and the function. This could happen because of extreme changes of acidity or pH or heat. It often permanently destroys the proteins, and they are not recoverable, and our body and our survival depends on taking the time and having the nutrients available to replace those lost proteins. So that's part of staying alive when our body is exposed to conditions that stress it out. We can talk about heat shock proteins. They combat denaturation. This is a group of several different kinds of proteins that your body makes in responses to stress. It was first found by heating up cells, so they called it heat shock. But it can also happen from other kinds of stress, for example, the presence of toxic heavy metals or changes in pH. This group of proteins helps repair and support the building of new proteins. And so it repairs our body in molecular ways, which gives an interesting reason for exercise. Besides just building up your muscles, exercise is a form of physical stress that improves your overall health by encouraging your body to make these heat shock proteins that start fixing up places where your body has been stressed out. Now we can talk about protein functions. Already, we've talked about what proteins are made of, the amino acids, and we talked about the levels of structure of proteins. Now we can talk about the different jobs they do. One of the most important jobs proteins can do is called catalysis, which is speeding up chemical reactions. The things that do catalysis can be called a catalyst in the subject of chemistry. It can occur outside of life. Catalysts can happen, for example, in the catalytic converter of a car, where it speeds up the breakdown of pollution from internal combustion engines. Now, catalytic converters work using platinum, which is a valuable metal, which makes them a valuable part of the automobile. But that's a place where the speeding up of a chemical reaction happens outside of life. Within life, catalysis happens by a special group of proteins called enzymes. Enzymes are proteins. There's a naming convention for enzymes. They end in the suffix ase. If you see a chemical in your readings or studies and it ends in the name ase, that's a clue to you that it's a kind of an enzyme. So everything that applies to proteins also applies to enzymes, but they also have this special naming convention. Now, substrates are the chemicals that are bonded together or broken up by enzymes. So in this diagram, a circle and triangle shaped substrate is one chemical. When it arrives at the active site of the enzyme will change shape, and that shape change will cause a chemical reaction, breaking apart these two parts of the substrate. So it can break things apart, or it could put things together, or transfer part of one molecule to another. There are thousands of enzymes catalyzing specific reactions throughout all living things. The enzymes facilitate or help the reaction happen. They are not used up by that. So enzymes are reusable. They're also specific to different substrates. So there are thousands and thousands of enzymes out there. Let me reemphasize this because enzymes are so important. See what's involved with the proteins known as enzymes. They have substrates, active sites, specificity, and reusability. This connects back with another trait of all living things in that enzymes drive our metabolism. The building up and breaking down of the nutrients needed for life happens in part because of enzymes. Another factor or a topic about enzymes is that they can be inhibited. They can be slowed down or made to stop, or their working can be blocked. This happens by chemicals that mess with the active site of the enzyme, Without the active site, the substrate can't fit to the enzyme. Maybe the best known group of enzyme inhibitors are known as the serotonin selective reuptake inhibitors, SSRIs. In the diagram here we can see two nerve cells meeting, and serotonin is a neurotransmitter that sends signals from one nerve to another nerve. And so the serotonin is released and the other nerve needs to receive it, and it causes changes in our nervous system. But SSRIs are forms of medicine, I guess you could call them, or treatments, that inhibit the reuptake of the serotonin. And so they slow the reabsorption of the serotonin. And so instead of getting absorbed back and stopping the sending of the signal, the SSRI makes sure that a bunch of serotonin sticks around, and so it strengthens the signal that serotonin is trying to send. That helps for people who do not have as much serotonin or have trouble processing it. And so people frequently know or understand the use and purpose of SSRIs. There are many other protein functions. They are part of the structure and the building of living things. For example, collagen is the most abundant protein in mammals. It's a quarter to a third of the protein content in the living body of a mammal. It's in the cartilage, bones, tendons, ligaments, and skin. Basically, collagen keeps the stretchy parts of us stretchy. It makes us more elastic where we need to be elastic. Or keratin is a protein made by keratinocytes. The word site is a clue that we're talking about a cell. And it's convenient, the name of the cell reflects the job it does. It makes keratin, so it's a keratinocyte. And that's found in scales, hair, feathers, nails, horns, claws, hooves, those parts of living things, including the outer skin of mammals. So those are two proteins involved in structure. Other proteins are involved in communication. We've already talked about hormones such as insulin and oxytocin, but there are also growth hormones made of proteins. Now, not all hormones are made out of proteins, but there is a group. So these proteins function in sending communication throughout the body. There are also proteins that make up the receptors to receive the signals. Bodies can be described as locks and keys, where some chemicals are signals, and parts of our bodies have receptors to receive and send those signals along to cause changes. Whether those changes are your muscles contracting, or the things that lead to your digestion or growth, or changes in where you're putting cells and how your body is put together, it's all a system of locks and keys. And for proteins, the shape of those locks and keys is based in the structure of the amino acids. Proteins have functions including membrane transport. They can move substances across cell membranes, whether by being a channel, and so an open tunnel into a protein, or by taking in one thing at a time, or letting things out from a cell one molecule at a time. Proteins function in our self-recognition, in our protection of our body. So our immune system has antibodies, and antibodies are a kind of protein. And so how do you know what makes up your body? It comes from the proteins in the immune system. Proteins are part of muscle contraction. The two proteins called actin and myosin pull against each other at the molecular level in muscle cells, and then thousands and millions of these lead to full muscle contraction, and so our bodies can move based on proteins. Proteins function as part of cell adhesion, and so they're a kind of a glue that holds one cell next to each other in the area called the extracellular matrix. You can think of our body as organized by layers on top of layers and compartments built within compartments. And so the bodies of all living things are built by and around proteins. In the world of our diet, we can talk about sources of protein such as animal sources, meats, dairy, eggs, fish. We can talk about plant sources such as soy, legumes, nuts, and seeds. And there are processed sources like casein or whey, and whey protein is a processed powdered source. But we can also reflect on the wider world. There are groups of people who get the major source of protein by eating insects. And, that might be different from what you're used to. But proteins are such an important part of our diet that people find ways to acquire those nutrients. Now here's a large graphic organizer, and it shows all the different things we talked about with proteins here. What about amino acids? How are they different? How are they put together? What are the structures of protein, and what words are involved with that? What functions do proteins do? And what about enzymes, that special group of proteins? So we can start to form a web of knowledge because all of these topics relate back to core ideas of biology and what it means to be alive. We talked about how proteins affect evolution, how they affect cells, how they affect homeostasis, how they affect our metabolism. And so we can really see some of the many jobs proteins do as a way to understand life. And so altogether, this has been our lecture on the biomolecules known as proteins. There's hemoglobin again. I hope this helped. Thanks for watching. Bye.